Some skin cells on the thorny dragon will not express the phenotype of a thorn. We're going to look at a developmental network that will have mutations that could hinder this, and we're going to look at that. So we will have two options, one being a thorn on the cell, an exaggerated cell shown here, or it'll be thornless. The first developmental network we'll see is the wild type. So S is able to stimulate P, which then it inhibits I. So I is not able to inhibit K. K then is able to stimulate E, which then is able to transcribe the phenotype of the thorn. So that's what that is. The thorn is able to be seen. The first mutation is going to be a mutant P. So we will throw the X on the P since it's mutated. We'll notice that even though that's mutated, S has no effect on it. So P's hindering I as a wild type, but now it can't inhibit I. So I then is able to inhibit K, which then is not able, because it's inhibited, to stimulate E. At that point, we get no transcription of this phenotype, and we now have a thornless thorny dragon. Next, we will have a mutant I. So we'll take that off, put it on I. At this point, K is not inhibited, so that's great for the thorn. We'll see the K is then able to stimulate E, and that's able to stimulate transcription, and we will get this. We will get the phenotype of the thorn. The next one is going to be a mutant I and E. So we will add the X onto the E. So we can forget about everything here because E is being inhibited in mutant. So we are going to not get the thorn. So we will now have a thornless cell. The final one is going to be a mutant S and K. So we will take this off of there, throw it on the S, and now we will have to inhibit and mutate K. So K is not able to stimulate E. So that means that E is not able to allow transcription to occur, and we will stay with our thornless dragon. So the epigenome is one way that this can happen by not changing the genotype or the DNA sequence of any of the cells, because they're all the same. So methylation is an effect that can happen, and methyl group could attach, or multiple methyl groups could attach to the DNA and tighten the coil. At that point, transcription is going to be inhibited. So we could look at possibly like the P mutant being inhibited with methylation, and that will then change the whole developmental network.